For our next installment of this neuroscience series, we're going to look at the activity of neurons. So the key concepts that we're going to be talking about here are basically, what are neurons and what are the supporting cells, those glial cells? How do neurons operate? By now you guys are pretty expert in this, but I'll go through this again. And then how neurons communicate with each other at the synapses. So we look at the nervous system as being critical to all organisms, but particularly to organisms that have more sort of advanced structures, because we need ways of kind of controlling and communicating information very rapidly within our bodies for lots of reasons, including for sort of the basic primitive ones of avoiding danger and protecting ourselves. Mammalian neurons are super fast, and this number, 20 to 100 meters per second, that's a pretty big range. So part of the difference between how quickly these signals transmit are both the length of the neuron, whether or not that neuron is myelinated, and also how thick the neuron is. So some neurons have much thicker axons than others. And in general, the ones that have thick axons and are myelinated are the fast ones. So we'll see that again coming up. We've seen these cells before. Here's some of the different cells that are supporting cells of neurons, the oligodendrocytes in the central nervous system. Notice this one single cell has these processes that are sticking out and wrapping around, forming myelin on this neuron. We also have some other cells here that probably you can't see too well because my face is in the way. There's microglia, and here we have the ependymal cells that are part of the connection with the astrocytes in forming this blood-brain barrier and connecting neurons with the bloodstream, so getting nutrients, etc. We know that neurons of all of these cells, neurons are the ones that we call excitable. These are the ones that can conduct electricity, and in fact do more than that because they do electrochemical signaling. Other cells that you see in here, that's not what they do, but they definitely are huge supporters of neurons. And in fact, there are more glial cells or neuroglia than even the neurons themselves. So basic structure of a neuron, you've done this a million times. Here you can see why I like this picture though, is it's showing that these dendrites that are extending out of the soma or cell body can be quite long. These processes can be very long. Sometimes they almost look like an axon themselves. But this is the receiving end of the neuron. Here's the axon, this one unmyelinated. And then we get down here to an axon terminal, which in this case there'd be a little bit of a synapse here. We just can't see it. That's going to be a connector between this neuron and this postsynaptic cell or postsynaptic neuron. We know that neurons are specialists in this conduction of electric signals, and that in order for a neuron to operate, it has to have some point of connection at the end, because neurons only do very short distance signaling. Now, unlike the hormones or the neurohormones that we looked at before, neurons are much more immediate in how they signal. So, and we've talked about all of this. We see down here this picture of a neuron. We're assuming an action potential here, but then we know that it has to get converted into a chemical signal before it goes to the next neuron. So hence the electrochemical signaling. Neurons innervate. So this term innervate has to do with what cells the neuron is actually contacting or communicating with. And that in the peripheral nervous system, a lot of times we talk about neurons as just being nerves, which are bundles of these axons moving in the same direction, maybe activating some muscle. But in the brain, when we talk about that, we're generally looking at things we call tracts. So when we talk about a tract, we're usually talking about the same thing, but a bunch of neurons going in the same direction, so we're sort of following the bundle of axons that are moving somewhere. So we have a lot of things that these glial cells can do, some of which we haven't gotten into any great detail on. One of them, which is really interesting, is that they actually help baby neurons, newborn neurons, go to where they need to go during embryonic development. They actually kind of create a scaffolding that neurons are able to move up and go into the proper layer that they would be in the cerebral cortex. They do some other things here too. You can take a look at this again and ask me questions if you have them before. Feel free to email me if you do. Here's some of the classic ones we've talked about and some of the details with them. Remember that oligodendrocytes basically they have a counterpart in the peripheral nervous system. We call them Schwann cells. The difference between these two, they both form myelin, but the Schwann cells do it by individual cells wrapping around. So on this picture here, you can see this is a single Schwann cell. It's actually, here's the axon on the inside. Here's the cell body, where this Schwann cell would be if we were to unwrap it. You'd see it there. And so it's forming this myelin in the peripheral nervous system. Here's another one, and here's another one. So each of these individually are Schwann cells. We talk a lot about how electrical current flows. We talk about voltage and membrane potentials. And this is just a little bit of a refresher on that. This idea that when you think of a current, you kind of think of a river. So you think about how things are flowing. So a current is that movement of electric charge that takes place. And it's based on the ions, the sodium ions coming in and then the potassium coming out. So we'll look at that in a minute. But voltage is this sort of measurement of the difference across the membrane, how much of a positive charge or a negative charge there is at any given place in time. And when we look at this voltage, that's what's going to produce the current, right? So we need to have a difference in this electrical potential difference, right? So we say membrane potential when we're talking about this difference that exists across the membrane. So we talk resting potential and action potential and just general, what's threshold and all of those things. So that's all referring to this. This is a great picture because it shows you, it sort of sets up, we've had this already, to show you what, once a neuron has sort of its basic setup done, in other words, the sodium-potassium pump has produced this, you see how much sodium is outside versus potassium. So a lot of sodium outside, very little potassium, and very few proteins, a few calcium. Chloride we've kind of ignored, but we're certainly going to get into more later, but very important too, because remember, chloride will hyperpolarize the neuron if it goes in, so it has that effect of making the interior more negative. So we look at that with GABA, we look at that with glycine, when it's inhibiting activity. But this is just setting up sort of what we normally see. So sodium concentrations are low inside, but potassium's high. Calcium extremely low, and a lot of proteins. But remember, these guys are the big ones that don't leave. So they're going to maintain that negative charge inside the neuron. This is just a way of measuring it. We've seen that before, just looking at what we call resting potential. Sodium-potassium pump, we know that always moves 3 sodium out for every 2 potassium it brings back in, and so it creates this gradient where you have far more positive charge out and a lot more negative charge in, even though we do have potassium is positive, but it's also in with a lot of other negative anions. Talk about potassium, though, is one of the ions that has this ability to sort of leak out, so through those leak channels. And remember that potassium would leak out if it could, because it's high concentration inside, so it'll keep leaking out. But at some point, as more and more positive charges from potassium go out, that's actually going to repel some of them from continuing to go out at the same rate. So it reaches its own equilibrium, where there's really no net movement of potassium anymore. And that actually is what eventually we would call resting potential. Here's that electrochemical idea, the electrochemical gradient. You move me out of the way again. There we go. So you can kind of see this. It's two things happening. One has to do with the concentration of an ion, and the other has to do with what's the electrical effect. So are we positive, are we negative, and how does that sort of either it propels certain things into the neuron or out of the neuron. All right. So here's some receptors. Ionotropic, remember we looked at these ones that are the ones that actually have a channel, and so when some stimulus lands on them, like a ligand lands on it and opens this channel, then we simply have whatever ion is going to be able to flow in, mostly. And then there's also a different type, the ones that are metabotropic, where the stimulus doesn't open a channel, but it does activate a bunch of stuff internally. The second messenger system, we talk about G protein coupled systems. It just means coupling means these are associated. And then once this gets activated, then it goes on to activate other things, sometimes then opening one of these channels. So it's kind of they call it a second messenger system. So ligand-gated channels are going to open on the receptors on the dendritic side. Most ion channels have a gate, and that's why we talked about why potassium leak channels are kind of special, and that why this effect actually does determine the resting membrane potential. Voltage-gated channels require a certain voltage in order to open or to close, and that's going to change due to either an influx, meaning coming in, or an efflux going out of certain ions. Here's the different just views of different channels. Not much to say about that. I don't really want to talk about the stretch-gated channels, and that is kind of it. So I'm going to stop there, and then I'm going to go into the action potential.